Hi, my name is Jose Bonilla Gomez, fish biologist for U.S. Fish and Wildlife Service and Alpina, and actual chair of the Coregonic Working Group, and I'm going to present the status report of the Lake Huron Restoration Project in Saginaw Bay. Before I'm moving forward, I would like to say thank you to all the partners involved in this multi-agency effort, which included federal and state agencies and also tribal agencies. In this presentation, I'm going to give you some history and background of the project. I'm going to talk about the stocking releases, the monitoring and evaluation that we're doing with the stockfish, and also the result we have up to date, what we're learning, and we're looking forward in this year and the next years. Related to the geographic distribution of Cisco and Lake Huron, I can say this historically, most of the Cisco harvest and Lake Huron came from Saginaw Bay area, and the current distribution of Cisco is limited to the northern Lake Huron, specifically the National Island, North Channel, and the Georgian Bay. And recent studies say the Cisco are no longer observed in Saginaw Bay and is rarely observed in the main basin. Related to the Cisco restoration study designs, back to 2007 when the Lake Huron Technical Committee drafted a document with a strategy and options for promoting the rehabilitation of Cisco and Lake Huron, where it concludes that Cisco rehabilitation will have tangible benefits for the ecosystems through a much needed diversification of the prey fish community, and also outlines a schedule of stocking and multi-gear evaluation methods to access the survival and reproduction of the Cisco hatchery. So this study design will include a Cisco gamete collection from northern Lake Huron, specifically Lake Chenot Islands and Drummond Islands, and then send it to Jordan River National Hatchery, where they reach the specific size, and then stock in Saginaw Bay. Saginaw Bay was chosen because it was good access to the main basin and is historically spawning sites. As I mentioned before, the Cisco gamete collection came from the northern Lake Huron because the need for genetic conservation is an important component in any fisheries restoration effort, and the importation of Cisco gametes from outside of Lake Huron for hatchery production would like introduce traits and behavior and cultural progeny that are not consistent with existing wild stocks. Specifically, Fish and Wildlife Service have been doing a great effort collecting those gametes from Lake Chenot Island and Drummond Island, and as you see here, those efforts have been producing a lot of eggs, enough to cover the stockings in Saginaw Bay. After several years of Cisco gamete collection in northern Lake Huron, we have enough data to describe or characterize the Cisco gamete collection in those areas. Usually the gill netting start prior to the spawn. It concludes when we have enough pairs to satisfy this proportion of eggs. That's usually around October the 27th to December the 6th. And we have been evaluating the CPU by photoperiod, temperatures, and moon phase. So that gives us an idea when exactly is the good time to head in there and collect those parents to extract the gamete. Here, for example, we can see with the information we have when exactly happened the spawning in the northern Lake Huron, specifically those areas we're trying to get the gamete collection, and we have been doing some general additive model to examine when that is or estimate when that peak happened. We can say that across all the information across all the year we have, we can say it usually happens around November the 10th. So that gives us a better perspective when heading there again and trying to collect the spawn and running. Here, for example, we know that temperature plays a significant role in determining the Cisco catch rates. And with the data and the information we have, we were able to do a logistic regression to estimate when those parts spawn by temperature. And at least the 50% of the population and the data we have, that happened around 7.7 Celsius. This is a good information that represents the biology of the species and allows to us to coordinate and make logistics heading to the field in the next years too. So after those Cisco reach a specific size and the temperature is secure, we release them in Saginaw Bay. And this is a big effort of coordination and logistics for transportation the Cisco from the hatchery to Saginaw Bay. And we have great help from the Jordan River National Hatcheries and also the crew from the Spencer Berry. Here you can see the Cisco stocking in Saginaw Bay, and I need to mention that stocking happens in two events, one in the spring and another in the fall. And today we are over 7 million fingerling Cisco stock in Saginaw Bay. And you can see in 2018 was the first year that we were able to stock fish, but that happened only in the fall because of logistic issues. In 2021 and 2022, we were able to stock 500,000 fish in both the spring and the fall. And also 2023 and 2024 have been good years for the program with over 1.2 million Cisco stock in offshore waters of Lake Huron. So what happened after the stocking in Saginaw Bay? And basically we're doing a monitoring and evaluation of all life stage of Cisco since eggs to spawning, and the evaluation was divided in two main components. One is detecting natural production, just to answer are the stock Cisco successful reproducing in Saginaw Bay, and also evaluating the survival and dispersal of stockfish, basically to answer how well are Cisco surviving, moving, and growing in Saginaw Bay. With respect to detecting natural production, immediately the ice is out in Saginaw Bay, we head in there and during 6 weeks we do larval tows using a randomized depth stratification approach using a Newton nets, towing with the wind at 2 miles per hour. And after that we do 4 more weeks of beach sailing to try to catch some Cisco. After several years of larval tows in Saginaw Bay, we have some results to show. And here you can see the larval density in Saginaw Bay. Larval density have very low in those sites we have been doing larval tows. But unfortunately, no Cisco have been detected based on morphologic, meristic, and genetics. We have some genetics results, and mostly they are lake white fish, and we haven't detected Cisco, but in other areas like Thunder Bay areas. To answer if Cisco returned to Saginaw Bay to spawn and stayed there, we have been doing surveys through juvenile surveys and spawning survey to detect Cisco returning to Saginaw Bay area. To answer the question if the Cisco basically survived and returned to spawn in Saginaw Bay, uh, we have some results to share. We have some Cisco catch in Saginaw Bay in 2022 and 2023. Here you can see the results of the gill netting effort we did in Saginaw Bay in 2022 and 2023 during the spawning survey. And you can see we found some Cisco close to the Oak Grade areas and south of Caseville. After we have those Cisco back to the lab, we do a OTC analysis. The OTC is a fluorescent mark and the hard structure of the fish that is provided by in the food at the hatchery, and where they provide one time to make a single mark representing the spring release and two marks representing two rings for the fall release. And that's why we can determine when it's a spring or fall release and it is a hatchery with OTC present. If it's no OTC present in the vertebrate, we can say that it's a wild fish. Here you can see where we found fish with OTC marks in Saginaw Bay. And also here you can see some picture of the fish that we catch. And the top one, it's a male hatchery with one ring and at the vertebra. And the other one, it's a wild when you can see any ring or OTC mark present in the vertebra. So up to date, we have 62 fish with OTC marks under UV lights in the lab. So sometimes we have some troubles at the lab reading the OTC. Sometimes the OTC marks are very hard to look at. And here you can see on the top fish, we have a Cisco with a very low OTC detect the vertebra. And at the bottom, you can see another Cisco with basically no OTC marks at all in the vertebra. So here is when we need to genetic confirmation to evaluate if the OTC is detected or is this or not, or is basically a wild one. Here you can see a table of the samples we sent for genetic confirmation, and we back from some results of confirming that some of them with low OTC are from hatchery parents, and we have another interesting results where we have back samples that was more related to genetic strain from Bruce Peninsula and confirmed that it could be a wild fish, and this will be a remarkable result, and we suggest that the wild fish follow the stockfish that were imprinted back to spawn. That is good news for us. So we have results from hatchery and also wild returning to Saginaw Bay. Here you can see the genetic confirmation of results provided to us from Amanda Atkins from the Great Lakes Science Centers, when basically related the population reference from our samples from Saginaw Bay, and confirming that we have some samples wild from Bruce Peninsula and other samples confirming they are from hatchery parents. So that is good results up to date. We have some, again, proof through genetics that we have Cisco's wild returning and also hatchery returning to spawn in Saginaw Bay. Another interesting results that we can share here, and Dave Fielder from Michigan and I can provide more details on that. Stochastic population model was developed to understand the potential trends and dynamic of reproduction population. Have you see here, there's a flow diagram demonstrating the age-based stage and transitions that serve as the framework of Great Lakes Cisco restoration simulation model. Those models, we put some inputs in the population like survival rates, imprinting, fecundity, stocking rates to get some of the results. Here you can see some of the results, and it's this baseline trajectory, and this model is based in simulation of 30 years. Stocking happened for the first year and 5,000 total simulation and a sex ratio of 1:1 in the model. So I can say that this arrow is showing what exactly is happening right now at the 6-year review, and we have some wild returning to Saginaw Bay. We're still providing some of the hatchery stocking in the system, and basically on the current parameter estimate, Cisco restoration via reintroduction is practical and has a reasonable likelihood of success. Also, I can say the initial simulation have demonstrated the importance of adult survival and the H0 survival rates in Saginaw Bay. In the previous slide, we have provided results of information that Cisco survived and returned to spawn in Saginaw Bay. But to confirm if those spawn produce eggs and that becomes part of the larva eventually, USGS is co-leading a sub-project to analyze to find eggs of Cisco in Saginaw Bay. Those locations have been close to the stocking size and also close to where we have been catch some spawners in gill nettings. Here you can see some of the results of eggs found in Saginaw Bay. You can see that there was lake whitefish, non-whitefish corrigoning, non-corrigonings, and other species. Here you can see that those non-whitefish corrigoning eggs were sampled near Cisco stocking sites we're assuming that they could be Cisco X, and we're waiting for the double genetic confirmation, and more details can be provided by Darrell Homeroot from the Great Lakes Science Center. To continue monitoring Cisco after being released in Saginaw Bay, Todd Hayden is leading a project evaluating the post-stocking Cisco mortality with acoustic telemetry in Saginaw Bay. And last September, 100 hatchery Cisco were tagged in partnership with Jordan River National Hatchery, USGS, and Fish and Wildlife Service. And those Cisco tags were released in October during the fall stocking in Saginaw Bay, and we're waiting for results this year to see what happened with those Cisco in Saginaw Bay. Of course, this is effort that needs a lot of help, and to evaluating the survival stockfish, we need help from different agencies or other institutions, and we are able to receive those peduncles from Cisco that have been caught in Saginaw Bay area and to analyze the OTC. So we're welcome to receive all those samples for reference. Through those all 6 years, we have been learning a lot, and there are some positive results. As you see here, a picture that is from the Gammy collection 2017 and another Cisco in 2023 from Saginaw Bay. Those are 6 years after, and those could be the first generation of those Cisco from Northern Lake Huron population. Of course, there is still much work to be done to answer the question. There are stock Cisco surviving and spawning in Saginaw Bay, but for now we can say the Cisco are in fact surviving and spawning in Saginaw Bay. We are moving in the right direction toward a feasible restatement of the self-sustaining population of Cisco, and the Cisco Restoration Project is helping us to make those goals. There's a lot more great work happening this year. In the fall, we're going to have another gamete collection in Lechino Island, another fall spawning sorb in Saginaw Bay. We're going to continue trying to find eggs of Cisco's in Saginaw Bay, and there's a lot of more work happening this year. Another juvenile Corregoni gillnetting survey, the Cisco dispersal and survival via telemetry, the effects of the UV and the larval production and density in Saginaw Bay, and developing a prediction for Saginaw Bay restoration project. And that's all I have for you. If you have any question, please email me and I get you back those answers as soon as I can. Or if anybody in the audience that from the Corregoni Working Group can answer some of the questions, I think they are very welcome to answer. Thank you very much for your time.